Hello everyone, myself Manish Kumar Dubey. I am faculty at Galgotia University at School of Basic and Applied Sciences, Department of Life Sciences. And today we are going to discuss about the topic cell theory and cell cycling. And this topic is covered under the course cell biology. Now before going into the detail of our topic, the objective of today's lecture is to understand the cell theory and to understand the process of cell cycle. Now, before going into detail of cell theory, let us first explore the historical background regarding the cell theory. The term cell was first of all used by Robert Hooke. He was an English scientist and he first of all described the or given the term cell in the year 1665. Actually, he used a very simple microscope to observe the cork plant samples under the microscope, and in this cork plant sample he found the hollow cavity which he called the cell. But actually these hollow cavities were not the cell, but it was actually the cell wall, and he published his drawing in the book called Micrographia. And here in the picture as well we can see the drawing of Robert Hooke, which he claimed that it was a cell. Now, but the actual discovery of cell is credited to Antony von Leeuwenhoek. Antony von Leeuwenhoek was a Dutch merchant, and he started building or means gathering the piece of lenses to construct a simple microscope in the early 60s or in the 17th century. And because of various microscope he constructed during that period of time, he is regarded as father of microbiology. And even the today microscope which we are using is mainly based on the design that is given by Antony von Leeuwenhoek. And it was Antony von Leeuwenhoek who first of all discovered single-celled organism, and he called this single-celled organism animalcules. And mainly he observed the bacterial cell, protozoa cell, and various other cells like mother muscle cells, fiber cells, and blood cell flow through his microscope. And because of his observation we came to know about the unicellular prokaryotes and what is the cell. The work of Antony von Leeuwenhoek and Robert Hooke paved the way for the development of cell theory. And in the development of cell theory, all the scientists who worked in the development of cell theory were from Germany during that time. And the cell theory was get proposed in the year 1838 to the year 1839. And the two main scientists who get credited for proposing the cell theory were Seddon and Schwann. In this, the Seddon was a botanist while the Schwann was a zoologist. So Seddon mainly observed that all the plant cells they are made up of cells, whereas all the animals were made up of uh, cells. It was proposed by the Schwann, and the concept that all living organism, whether it is bacteria, whether it is plant, whether it is fungi, or whether it is any animal, they are composed of cell. It is called as cell theory. And this concept is later on extended by Virchow, who proposed that omni cellula a cellula. That means all cell come from the pre-existing cell. So all this discovery later led, led to the development of cell theory. Now what are the three main things that the cell theory state? So the first thing is that all living things are composed of cell. That means whether it is unicellular organism or whether it is multicellular organism, all the organism they are composed of cell. Now the second important point in cell theory is cell are basic unit of structure and function in living things. Means different type of cell, whether they look alike or functionally they are different. They have certain kind, they can have alike function or they can have dissimilar function inside an organism. Now the third important point of cell theory is new cell are produced from the pre-existing cell. Means the pre-existing cell can reproduce to give rise to a new cell. So these are three very important points that are proposed in a cell theory, and the point over which the cell theory is based on. Now there are some exceptions to the cell theory. So the most important exception to the cell theory is viruses. We know that the viruses is considered as a means boundary between living and non-living organism, and most of the viruses are obligate parasites. That means they are subcellular in nature and they need a host for reproduction. Similarly, there are many bacteria like Rickettsia and Chlamydia. They can be also considered as exception to cell theory. Another example of exception to cell theory is Mycobacterium, Mycobacterium leprae or Mycobacterium tuberculosis. They are also obligate parasites. That's why they can also be considered as exception to cell theory, because all these three basic points that are proposed in the cell theory, they are not get fitted over these organisms. That's why they are kept as an exception to cell theory. Now the next important topic which we are going to cover in today's lecture is cell cycle. So before understanding the process of cell cycle, we have to understand what is cell division. So according to cell theory, we know that all the cells, they get arise from the pre-existing cell by a process called cell division, or the phenomena by which daughter cell is produced from pre-existing cell or the parent cell. It is called as cell division. And why do the cell need to divide? To maintain the nucleoplasmic to cytoplasmic ratio or karyoplasmic ratio, so that the ratio will get distributed or it will remain maintained throughout its progeny. Now first of all we have to understand what is cell cycle. So in a population of dividing cell, whether inside the body or in a culture dish, each cell passes through a series of defined stages which constitute the cell cycle. And the cell cycle can be divided into two major phases based on the cellular activity readily visible with a light microscope. So what are these two major phases of cell cycle? One is M phase, another one is interphase. M phase is also called as mitotic phase, or in the cell that is undergoing meiosis, it is also called as meiotic phase. Now first of all let's focus over the interphase. So interphase is also called as resting phase, but actually it is not a resting phase, because the cell is most metabolically active during this phase, and it is mainly composed of three subphases. And what are these three subphases in interphase? One is G1 phase, then S phase, then G2 phase, and it is much longer. The interphase is much longer as compared to the mitotic phase or M phase, because it takes around hours to few days to few weeks to complete this interphase, and it is the period between the cell division, or it is the time when the cell grows and enlarges before getting divided in the M phases. So after the cell prepare itself for the division in the interphase, then it enters the M phase. So M phase is actually the dividing phase, but it is of very short duration. That means it lasts for only few hours, and it includes two processes. First is mitosis, during which duplicated chromosomes are separated into two nuclei, and later on there is karyokinesis, during which the entire cell divides into two daughter cells. And the karyokinesis is by the process can be happen by the process called cell furrow formation or cell plate formation in plant cell and animal cell. Now, although the M phase is a period when the content of cell is actually divided, numerous preparation for the upcoming mitosis occur during the interphase, that also include replication of cell DNA. So let's see the subphases of interphases into detail. So the interphases, first phase of interphase is G1 phase, that occur after the completion of cytokinesis. So G1 phase is also called post-mitotic phase or pre-DNA synthesis phase of first gap period or the first growth period. and it is the highly metabolic phase of the cell in which the synthesis of protein, RNA and ribosome take place. Normally it takes around 30 to 40% of total cell division cycle, and the division of cell, whether it will undergo during the cell process of cell division or it will not, it will occur during this phase of cell cycle. Now this phase of cell cycle, it is followed by another phase that is called as S phase or DNA synthesis phase. In this phase what happens, the DNA replication and the synthesis of histone protein mainly occur, and the DNA polymerase that is required for the replication of DNA, it becomes fully functional in this phase of cell cycle, and it requires a time that is around 30 to 40% of total cell cycle time. Now in this phase what happens, once the cell enters the S phase, then it will definitely undergo mitosis or cell division without any interruption. This phase is very important. Now after the completion of synthesis phase or DNA synthesis phase, S phase, the cell enters the G2 phase, and G2 phase is also known by pre-mitotic phase and post DNA synthesis phase or second gap or growth period phase, and it requires much less amount of time as compared to the G1 and S phase of the cell cycle. Now what happened during this phase of the cell cycle? The division of mitochondria, chloroplasts, centrioles and synthesis of other required protein that is necessary for the cell to undergo mitosis that occur in this phase of cell cycling. And one of the important thing, that is formation of protein that is required for spindle apparatus, that also occur during this phase of cell cycle. And after this phase what will happen, the cell will prepare itself to undergo the mitosis. So after the completion of all these phases that comprise the interphase, the cell will now enters the M phase, that is also called as mitotic phase, or it is also known as meiotic phase in the cells that undergoes meiosis, or it is the actual cell division phase. So as we have seen, this M phase, it is shortest as compared to the interphase. It do not take much time to get completed, and it was observed that Vasia feber or broad beans bacilla, here the interphase is around lasts for around 30 hours, whereas the mitotic phase or M phase, it lasts for few hours, around 1 hour. Now the cell cycle have three checkpoints which check the cell, or means which arrest the cell activity to before undergoing cell division. If they found there is a certain kind of error that happen during the cell cycling processes. So what are these checkpoint? First checkpoint, that is occur at the end of G1 phase, that is also called as cell growth checkpoint. Then another checkpoint that occur during the process of DNA synthesis, that is called as DNA synthesis checkpoint. And last phase that occur as a checkpoint is mitotic checkpoint. So let us discuss these checkpoints one by one into detail. So first checkpoint that occur is called as cell growth checkpoint. It occurs towards the end of G1 phase, and it checks whether the cell is big enough and have the has made proper protein for the synthesis phase or to carry out the DNA replication process. And if the cell is not prepared to undergo the DNA replication process, then what will happen? The cell will undergo the G0 phase, that is resting phase, in which cell become dormant, or later on then it will again get converted. G0 phase can again become active and get converted into G1 phase, in which cell start again preparing the protein that is necessary for the DNA replication process. Now once the cell passes the G1 phase then and enters the S phase. So in S phase what will happen? There occur another checkpoint that is called as DNA synthesis checkpoint, and in this checkpoint the cell check that whether the DNA has replicated correctly or not, and if there is the replication of DNA is not correct, or it have some means damage inside it, then what will happen? Total cell cycling process, it will get arrested and cell will stop uh, undergoing the other phases like G2 phase or M phase. And if all the process will happen correctly, then cell will enter the G2 phase, and after that it will pass to M phase. And in M phase, what will happen? The third mitotic checkpoint occur, and in this checkpoint what will happen? It will check that the cell have efficient component to undergo the process of mitosis or not, whether the various protein that is required for the synthesis of centrioles, or whether the all the organelles they get divided or not, or whether the cell have made all the protein that is required for the formation of spindle formation that occur or not, or if there is any disjunction that occur during this process or this M phase, then what will happen? Cell will arrest itself and it will not go towards the cytokinesis uh, phase. So overall these three checkpoint checks that whether the cell cycle is followed correctly or not. Now these are the references which will be used for